Okay, so we're still on chapter 11 for AP Biology, talking about cell communication. Previously, we talked about an overview of what cell communication was in the three parts: the reception, the transduction of the signal, and then ultimately the response. Again, we've covered as a separate topic just the types of receptors that are found on the cell surface that allows for that initial reception to occur. Those are the G proteins, the tyrosine kinases, and the ion channels. Today we're going to follow up with the transduction part of the cell communication process. So cell transduction is a cascade of reactions, so a series of reactions that relays the signal from the receptor to our target molecule. It's always multiple steps, and that happens so that we can amplify the signal, so we can make the signal bigger, we can allow it to reach more locations. It also provides for more opportunities to control the signal and the process, because if we shut down the process at any step along the way of the transduction, we can then halt the production of the response. Most of the time when we talk about signal transduction pathways, we'll be including something called a phosphorylation cascade. And as the name implies, we're going to be using ATP or GTP to be phosphorylating molecules. And then at each step in this pathway, we have to recognize that the signal, the reception, is transduced into a different form. And usually the different form is simply the conformational or shape change of a protein. Now, transduction. When we talk about the amplification or the regulation that happens, one of the words that gets thrown around is crosstalk. And what that does is that allows us to coordinate kind of incoming signals or incoming ligand molecules. So if you look in this diagram here, we have our signal molecule binding to our receptor in the membrane. So once the ligand is bound to the receptor, that will then activate or transduce into the activation of relay molecules within the cell. What they're trying to show here is that sometimes when we have the activation of these relay molecules, we can trigger one response, or we can trigger multiple responses using multiple relay molecules. The other process is that when we can have what's called crosstalk. And crosstalk is where when one signal is received versus a second signal, they're going to be amplifying and or inhibiting each other. So we can see in this diagram that the green ligand, once it binds to our signal molecule, we're creating the relay molecule that then can activate or inhibit a relay molecule for a separate communication that happens, which then triggers another response. The other thing that can happen, or the last thing if we look in this fourth box labeled D, is that a different receptor can lead to a different response. And so you can simply have the same molecule, but because it goes to a different receptor, we're going to trigger an entirely new process. So let's look at this. When we talk about this crosstalk or this amplification, more often than not, what those relay molecules are protein kinases, or they're activated by protein kinases. So a protein kinase is an enzyme, and it's an enzyme that specifically adds phosphates to other enzymes. Now, there's the enzyme that adds the phosphates, and then there's an enzyme that undoes the phosphate, which is called a phosphatase. Phosphatase enzymes remove the phosphate, which then deactivates, most oftentimes, deactivates the enzyme in the phosphorylation cascade. Sometimes by removing the phosphate, we actually can activate. It's a rare case, but it does happen. Now, when we look at a phosphorylation cascade, here is an example of what happens in the transduction process with those relay molecules. So if we look closely, here is our ligand signal molecule binding to the receptor in the cell membrane. We've now activated our relay molecule. This active relay molecule will then have our protein kinase become active. How do you do that? Well, you're going to phosphorylate another protein kinase, which will then trigger the activation or phosphorylation of another protein kinase, which will then eventually trigger the activation of a specific protein, which leads to the third step in communication, which is a cellular response. So here's our relay molecule activated activating our protein kinase. That active protein kinase, our first one, number 1, transfers the phosphate from ATP, which then phosphorylates protein kinase 2, which is now active. Protein kinase 2 is then going to phosphorylate protein kinase 3. This could happen 4, 5, 6, 7, 8 times, all of which are places in which we can regulate the communication, until eventually the final active protein kinase will phosphorylate our protein. That protein will then trigger the response within the cell. Now, again, we're going to talk about those phosphatases. Phosphatases are the proteins which remove the phosphate from the protein. As soon as you remove the phosphate from one of these active protein kinases, it becomes inactive until another signal is received that will then reactivate it. Now, not only are protein kinases involved in the process of transduction, there are also very small secondary messengers that can happen. These are things that are small, non-protein, water-soluble, and oftentimes they're things that are small compounds that can be broken down in water, for example, polar molecules. One of the best examples of a secondary messenger is calcium, and calcium plays a role in our, when we have our cell and it receives a signal, what's going to happen is we're going to pump calcium into our endoplasmic reticulum. We're going to pump calcium into our mitochondria. Why would we do that? Because that would then lower the amount of calcium inside the cell, changing the concentrations. That creates potential energy, which could be used to generate a cell process later. So this is one of the best examples of ion channels and secondary messengers. So the message is actually the calcium, or the concentration of the calcium. Another secondary messenger, which we'll become very comfortable with, is cyclic AMP. Cyclic AMP is actually a molecule made from ATP. So here's our big ATP molecule. It's actually not very big, but here's our ATP molecule. We're going to use an enzyme. And what the enzyme is going to do is it's going to remove 2 of the phosphates. We're going to add water, and we're going to be breaking some bonds. And if we compare, this is our AMP. So here's our cyclic AMP in the middle, and then here's our AMP. What did we do? You'll see that we are changing the phosphate group. So cyclic AMP is actually a secondary messenger that's made from ATP. So here is how do we make cyclic AMP. So here's the pathway that is the communication that says, let's make our cyclic AMP. Okay, so first things first, we're going to have our G protein receptor. Here's our ligand. It's going to come in and bind. Reception has occurred. We're then going to transduce the signal, and what we're going to do is we are going to be activating our G protein. Once G protein is active, it will then force the activation of our enzyme, which will then add a phosphate to or take away the phosphates for our cyclic AMP. Cyclic AMP then becomes the secondary messenger, which then activates a protein kinase A, which could then lead to another phosphorylation cascade. Protein kinase activating protein kinase, activating protein kinase, until we ultimately get our cellular response.